All right. Good morning. I'm going to get started with what we have for this week. So this is the syllabus for our lab portion. This week, tomorrow, lab practical number 2 is going to be due. We're going to complete looking at the senses. We're going to look at the sense of smell and the sense of taste. We're also going to get started with an overview of the skeleton and the skull. There's a lot of bones, and due to the complexity of what is occurring at the level of your skull, all of the nerves that are going in and out, all of the cranial nerves that are going in and out, all of the vasculature that is going in and out. There's going to also, you're gonna need basically complex piping, a complex infrastructure that will allow all of those lines of communication to go in and out. We're gonna look at all of that. All right, let's go to these chapters. All right, we're going to look at the two remaining special senses, olfaction and taste. We talked about the characteristic of special senses. Special senses will contain an organ that will facilitate the transduction of whatever signal they are monitoring. In this case, in the sense of olfaction, we are monitoring these volatile odorants that are in the air. Whenever you smell something, it's due to the physical interaction between an odorant and a receptor. Um, so we have our sense of smell has developed a lock and key mechanism to specific receptors. Um, there are substances that we're not able to detect um, through smell. For example, you heard that propane gas doesn't have a scent, and whatever we smell is added to it. That is because we do not have the receptor for that particular molecule. So whenever that molecule enters, comes in contact with our olfactory epithelium, it just doesn't trigger a receptor. There's not one that does. But once we add that odorant to the gas, we are able to detect the odorant, and therefore we do know intuitively what the scent of gas is. But once again, it's a lock and key mechanism, and this is the system that allows us to experience that. So at the olfactory cavity right here, you're going to have several olfactory nerves coming in contact. Um, the surface. Once we capture once the scent goes into our system, there's up here an olfactory epithelium, and that olfactory epithelium will be innervated by this olfactory nerve. Olfactory nerves will in turn go on synapse here at the olfactory bulb, and the olfactory bulb will send the signal to the central nervous system. Here you have some of the basic anatomy of the olfactory nerve. You have here different this bony structure called this meatuses and conches. But let's look at it in more detail at this at the olfactory epithelium. So here we're going to look at bones of the skull in a moment. Here you have the cribriform plate of the ethmoid bone. So you have this olfactory cavity, and within it you have these different these bony structures, this nasal concha. What the nasal concha does is it provides this provides this tortuous environment that circulates and turbinates the incoming air. That turbination allows more contact between these volatile odorants and the olfactory epithelium here, these olfactory neurons. So there is greater chance of the receptors within the olfactory neurons to come into contact with the odorants. Not only that, we are moist in that process. The cell is highly vascularized. That is why whenever we have a nosebleed, you see that it bleeds profusely, even though you don't. Sometimes there is an extensive damage done to the area. It's the mere rupture of these very superficial capillaries that are present. And the reason why they're present is because water is great at conducting heat. So whenever we breathe, especially during winter cold, this is more relevant. Whenever we breathe cold air, that those exposed capillaries are able to warm it before it reaches our lungs, and our lungs are very close to some vital organs, the heart, and the core of our structure. So we want to warm that. In that process, we're warming the air and we're hydrating it at the same time before it reaches the alveoli of our lungs. So here you have this is a closer look into the cell structure of the olfactory epithelium. The olfactory epithelium will contain the olfactory neurons present here. That the olfactory neurons contain this long dendrite that will branch into several olfactory cilia. So this we talked about cilia whenever we talked about this motile cilia present in the lungs. Present in your trachea and bronchioles, in order to perform this movement of debris out of the delicate structures of the alveoli. And we talk about olfactory cilia as well in the uterine tubes, once again creating this wave-like motion. Now here we have an olfactory cilia whose main role isn't really movement, but it is, they have receptors within the surface. This is where these odorants come into play. And each olfactory cilia makes one, each olfactory neuron makes one specific type of receptor. Let's say that we have a receptor that senses this citrusy smell. Only this neuron will have only that receptor for that citrus. It could be that several neurons will have it. It could be that this neuron has a citrus receptor, and then this neuron also has a receptor that will allow it to sense the citrus scent. But all together, they only have one, and perhaps this neuron has a version of it that allows us to send a specific type of citrus. So there we get some level of resolution. And then all of the neurons that have that citrusy response will converge within one glomeruli. So they all are responding to basically the same level of combustible response. It makes sense since they all are firing simultaneously. And from here, it would interact with a mitral cell that sends that signal through the olfactory tract to the central nervous system. What else do we have here? We have also supporting cells near the new supporting cells, and we have here stem cells. You notice that the olfactory epithelium has a mucus surface, and that's going to be provided by olfactory glands. So here, let's look at in this view, we're able to see the cilia, the sensory cilia, in the lumen of the nasal cavity. We're able to see. It's hard to tell the difference between supporting cells and olfactory sensory neurons. All of these nuclei pretty much look the same. We can almost hint that we're going to have the olfactory sensory neurons in this end, but it's not a rule of thumb that always follows. So it is challenging. I wouldn't expect you to tell the difference between a supporting cell and an olfactory sensory neuron based on this thing. On the apical layer, on the basal portion, this is the apical portion, the portion facing the lumen. On the basal portion, we will see stem cells, and you can see almost a line present here of stem cells. Further, we mentioned that whenever you see an opening, that is either a blood vessel or a gland, or a mere just rupture of the staining process. In this case, the opening is surrounded by this simple cuboidal epithelium. So we can almost infer, by that this is a gland. Excuse me. All right, so this is kind of a similar view. All right, this is the gland that we were talking about with the simple cuboidal epithelium present here. This so this is an opening, right? And the difference between this opening and this opening is that this is simple cuboidal epithelium, and this is simple squamous epithelium. Glands glands do not have simple squamous epithelium, but blood vessels do. Blood vessels do have simple squamous epithelium because they want exchange to occur. And the best epithelium for that is simple, it's not stratified, and it's squamous, it's a thin cell rather than a thick cuboidal or columnar cell. And here you can see a hint in the interior of red blood cells. This opening is hard to follow that logic. Oh, here. This opening right here, and it doesn't look like an opening, right? But you can see the red blood cells present here, and you can see the simple squamous epithelium surrounding it. So this is a blood vessel. Anyway, we did mention that there's a lot of vascularization within this olfactory epithelium. That is so that whenever air flows through it, this highly vascularized portion present here in the lamina propria, that highly vascularized area will hydrate and cool the air as it goes through the nasal cavity. All right, that is your olfactory epithelium. Now let's look at your gustatory sense, your taste. We're gonna focus especially on taste buds. Here you will have several types of papillae. These protuberances within the epithelium that they have a functional significance. At the back of the tongue, you're going to have these vallate papillae. They form a V-shaped structure at the back of your tongue. On the side of your tongue, there's this rugae area. These are going to be your foliate papillae. You would also have fungiform papillae, which look like goosebumps all throughout the surface of your tongue. These papillae, some of these papillae will have among them interspersed these taste buds present here. Here we also receive a view of the epiglottis. Whenever we swallow, the epiglottis is going to cover the trachea, because as you may notice, we can breathe and eat through the same opening, meaning that at some point, these openings need to diverge. One of them will go to the trachea and the respiratory system, and the other one through the esophagus and the digestive system. Whenever we eat, one of them, if we digest food and we don't close if we don't close a valve over the respiratory system, then that food might go into the respiratory system. This is where the epiglottis plays its role. Whenever we swallow, we raise your larynx, your voice box, and whenever we raise this, the epiglottis kind of closes like a lid, sealing the entrance to the respiratory system. Here we have a view also of palatine tonsils. Those are tonsils on each side of your oral cavity that whenever you have an infection, you'll see them enlarged. That means they're doing their job. And at the back of the tongue, you also have lingual tonsils, not as visible. But let's look at the taste buds. So here we have a vallate papillae, and this vallate papillae will contain, first of all, it will be formed of stratified squamous epithelium that's all throughout your tongue. We mentioned the best tissue for the best epithelium for protection is it's not gonna be simple. It's gonna be layered. It's gonna be stratified. And in order for us to pack more cell layers within the same area, um, the epithelium is going to be squamous. That way you can fit 500 layers of squamous instead of 50 layers of cuboidal, or maybe 20 layers of columnar. If you have less layers, then whenever a layer gets shed off, a huge portion of the epithelium goes. So that's the reason why it is squamous for protection. But within this epithelium, there are taste buds. These taste buds contain these gustatory epithelial cells with the specializations at their surface called gustatory hairs. This is a specialized. This is whenever we talk about the spatial senses, we talk about it. Whenever we talk about senses, either the signal triggers receptors directly into the nerve, into the dendrite of the neuron, or it triggers receptors within a specialized sensory cell. In this case, we have the specialized sensory cell that gets triggered. These gustatory epithelial cells, specifically gustatory hairs, specifically these specialized structures called gustatory hairs get triggered. And that this cell sends a signal to the dendrites of these particular neurons. These signals enter the taste bud through the taste pore. Here, this small opening present here will allow the signal to enter, and from there activate these gustatory epithelial cells. Here we can tell the difference between these gustatory epithelial cells and the basal epithelial cells, which will give rise to more gustatory epithelial cells. So those were the valate papillae. Here we have a view, and the valate papillae are fairly large in terms of size compared to the rest of the papillae. Here we have a view of foliate papillae present on the side of your mouth, and this foliate papillae will, as you can tell in the side of the mouth, there's this rugae present, this indentation, invaginations. This depicted here. The foliate papillae will have taste buds, as you can see here, taste buds present. Here's the taste pore through which these substances come in direct contact with receptors present in hair, in epithelial, in the hair area of gustatory epithelial cells. All right, any questions about the gustatory and respiratory system? Nope. All right, so let's jump into the skeleton. The skeleton and the muscular system, it's going to require a lot of root memorization. How it's like trying to learn a phone number. It's or trying to learn a long number. there's not much that can be explained about the number that will help you. We'll go through it, and that's what's going to help you the most. Is just like repetition. So I suggest once we go through the parts of the skeletal system that you immediately try and digest this material once again, either through the same medium, this PowerPoint presentation, or through different mediums. I would suggest the latter. Just choose different mediums to digest it, your textbook, um, Wikipedia, there's countless videos online about it. There's going to be this video. Um, all of those resources, but just like try to expose yourself in several times to the same material until then some of these names are not going to be familiar until the names start sounding familiar, and then once they're completely familiar, you will start assigning start assigning the name to its visual representation. But it is challenging. There's 206 bones, and we're not going to just learn those bones, but we're going to learn details about those bones. They have several structures that within each specific bone that are relevant to their function. So here we have, this is your stereotypical long bone present here. What is the role of bones? That is kind of like the first questions that we have to ask. Role of bones is going to serve in protection. They're going to have a chewy center that any carnivore can attest to, from which hematopoiesis occurs. This is where blood gets formed. They also serve not only in protection, but they serve in locomotion. So in order for locomotion to occur, what do we need to happen? We need muscles to attach to these bones so they can move them, so they can create these motions. So there's going to be, in the surface of each bone, there's going to be indentations, processes that are going to, in a way, what's the opposite of eroded? Because the more that specific muscle is used, the stronger or the larger that particular indentation or mark on the surface of the bone will become. And that is because the more pressure, it basically is a trigger to the bone saying, hey, we're using this a lot. So make sure you strengthen this particular connection. And it does. Bones are fairly dynamic in that sense. Calcium and phosphate reservoirs are within bones, and we need to have a homeostatic balance of those. Whenever we have low levels of calcium, we're going to release hormones that will basically resorb bone so we can get calcium into the circulation, so we can increase our blood calcium levels. And that is happening constantly. That's happening right now within your bones. We're resorbing bones. However, we're also depositing calcium whenever you get a meal, whenever you drink a tall glass of milk. There's calcium within your blood, and you're going to have cells called osteoblasts that are going to deposit that calcium. They're basically grabbing calcium from the blood and putting it in your bones, making them stronger. So this is a constant treadmill of calcium resorption and absorption and deposition that is occurring at your bones. And this constant remodeling of bones will be also will happen depending on how active those bones are, or how much a particular tendon pulls in a particular area of your bone. We're going to need more calcium in there, so we're going to deposit more calcium in that particular area. All right. So with that, we know why this long bone has a hollow medullary cavity. That's for the red pulp and these osteo or hematopoietic cells to reside. We also know that there is compact bone due to to provide strength for locomotion and structure. And here we have at each end, we have a spongy bone. And the spongy bone, it's going to be present in areas where there is, these bones are not going to exist in an island. They're not going to be isolated. In order for them to perform their function, they're going to pivot, they're going to join other bones. And the spongy portion help with kind of buffer some of these pressures that are going to occur between bones. And so that is why we have that here. We're going to, each long bone is going to contain one diaphysis and two epiphyses at each end. The epiphyses contain, the transition between epiphysis and diaphysis is marked by the epiphyseal line. The epiphyseal line is the area where cartilage is still being deposited, and cartilage can transition into bone, can be calcified. That is basically how bones grow. These fibroblasts, these chondroblasts produce cartilage. This cartilage gets calcified, basically, and that calcium becomes that becomes part of bone. Okay, today we're gonna focus on the axial skeleton, and this is a view of the axial skeleton, and later we're going to focus on the appendicular skeleton. So let's start with the skull, the cranium. Please become familiar with all of these bones. So here you have clavicle, scapula, those should be familiar, sternum, ribs, humerus, radius, ulna, with the radius connected to the thumb, and the ulna to your fifth finger. You also have here your pelvis containing your ilium, your ischium, and the pubic bone. So here you have your femur, tibia, fibula. Your hand and leg bones are divided into carpals at the wrist or at the ankle, tarsals. Then you have your metacarpals and metatarsals, which would be analogous to this area right here. And then you have your metacarpals, 3 metacarpals per finger, except for your thumb that contains 2. So it's like proximal, medial, and distal. This one only contains proximal and distal metacarpals. You can everything. So you have your carpal bones, metacarpal, carpal bones at the level of your wrist. There are 8 carpal bones. You have your metacarpals here, 1 through 5, one starting at the level of your thumb. Meta, you have your phalanges. So you have your phalanges present here. And your phalanges are going to be your proximal, medial, and distal. Once again, your thumb only doesn't contain the medial phalange. You can translate this knowledge into what happens at your feet through the tarsus, metatarsus, and phalange. So let's look at bones by their classification by their shape. We're gonna have long bones. We're going to have flat bones, irregular bones, and short bones. Your carpal and your carpals and tarsal bones are going to be examples of short bones. Irregular bones, here your vertebra. Flat bones, your cranial bones are going to be flat, as well as the sternum. And long bones are going to be present mostly in the appendicular skeleton, in your limbs. So for the most part, their name is fairly descriptive, what a flat, short, long, irregular bone is. All right, let's get to the thick of it. Excuse me. So let's focus on bone markings. Once again, these bone markings, they are there to reflect a particular function. Here we have projections. These are the sites where muscle and ligament attach. So muscles attach through tendons to bones. Ligaments are attachments between bone and bones. So an attachment between muscle and bone is going to be a tendon. An attachment between bone and bone is going to be a ligament. The end result of these attachments is pressure within the bone, right? Either pressure because of the muscle contracting, pulling on the bone, or these two bones moving in relationship to each other and kind of pulling on each other. This pressure is going to, this pull within the bone is going to cause the position of calcium to strengthen that bond between the ligament and the bone, between the tendon and the bone. This the position of calcium creates this surface marks, and that's what we're going to look at. These projections, these surface marks. So some are at tuberosity is one of these projections. Here in the pelvic bone, you're going to have a tuberosity. Crests are going to be also these projections. And here you have a large amount of muscles being, a large amount of tendons attaching to this particular crest in the pelvic bone, the ilium. So that's kind of why they're formed. That is why we're going over this. It's just to it kind of helps us understand some of the function of these bones. That bones are not there just to provide the structure. They're there also to play a pivotal role within locomotion, depending on where because muscles are fairly straightforward in their function. either contract or relax. They only move in one, skeletal muscles, they only move in one axis, the axis of contracting, they're either contracting or relaxing. They don't perform two-dimensional or three-dimensional functions. I guess I should say they don't perform three-dimensional movements. So how do we get this really complex range of motion? And that is really done because we have a large variety of muscles working in unison to create this sense of motion. But each individual muscle would only create this one-dimensional type of motion. The way that it does it is it attaches, if you have a muscle attached within, it has an origin and an insertion point. If both of them are within the same bone, whenever the contraction is, they would not create any sort of motion. They would just, imagine if I put a rubber band, I stretch it. Do I have a rubber band? Not readily. But imagine if I put a rubber band between this area of the pencil Imagine if I put a rubber band between this area of the pencil and this area of the pencil, and the rubber band is attached to the pencil, and I let go of, and I stretch it and I attach it and I let go, then the rubber band is just going to stay in place. Even though it wants to retract, it's not going to create any sort of movement because it's attached on the same place. Now, let's say that I staple the rubber band to this end, and then I staple the other end of the rubber band to this pen, to this part of the pen, and it's a stretch rubber band. I stretch it and I create that, and I create the insertion here. Then we let go. Whenever we let go, then the rubber band will pull on this pen, effectively creating this motion. And then, well, rubber bands cannot relax, but muscles can. They can contract and then relax. But I hope what you get out of here is how it is intuitive that each muscle has an origin in one bone and the insertion point on a different bone. Because that is what creates this motion. If it was within the same bone, you're not going to have any motion. So these origin and insertion points are going to create these processes. Then we're going to have these different projections that help form joints. And we're going to have depressions or openings that are usually for the passage of the piping of biological creatures, which is going to be nerves and the vascular components. There are also different openings. We're going to see in the cranial nerve, we're going to see fossas, which are like these cup-like indentations. We're going to see sinuses, which are going to be these openings within these cavities within bones. And we're going to see meatuses, which are going to be canals. For example, your auditory meatus. We need the nerves that, so here, whenever that air waves reach your tympanic membrane, eventually get translated through the ossicles into the cochlea that sends a signal to the vestibulocochlear nerve. Now, that signal of the vestibulocochlear nerve will go to the midbrain, the inferior collicular, before it goes to the thalamus. It eventually went from the exterior of our skull to the interior of to our brain, right? To the interior of it. It has to go through an opening within the skull. That opening is a canal. That canal is a meatus, this particular one, the auditory meatus. All right, let me share once again. Can you see my screen?
Yes.
Awesome, thank you. All right, so that's projections. We talk about between muscle and between tendons, ligaments, and bone, between bone and bone in the form of joints. Then we have openings or depressions. So let's take a closer look at these specific types of projections. You have tuberosities, you have crests, you have trochanters, you have lines. That's going to be for here you don't have a sample of a line, but within the tibia there are you have a line there, which is just like a protuberance in the form. It looks like a line, literally a line within the bone. And that's one of the easiest ones to see. You have tubercles, which are small processes. So what's the difference between tubercle and a trochanter? Is the size. Trochanter is going to be large, where a tubercle is going to be smaller in size. You have an epicondyle. What's an epicondyle? It's a projection above a condyle. What's a condyle? A condyle is an indentation within a joint that helps. Actually, it's so some of these joints are going to have an area that crests and an area that indents, and they're both going to have different names. The area that crests is going to be the condyle. Next to it, if there is a protuberance next to it, you're going to have an epicondyle. Now, what I want you to understand is you don't see these protuberances here, right? You don't see them in the middle of the bone. You see these protuberances right at the diaphysis, at the ends of bones. That is because of what we mentioned earlier. Whenever we create this, whenever there is an origin and an insertion point for a muscle, it's more efficient if you pull on the proximal end of that particular muscle, on the edges of that muscle, whereas compared to if you pull in the middle section of that muscle. So this particular muscle is attached to, I said muscle, bone. This particular, here you have the femur. We're going to have several muscles that go from the pelvis to the femur, as well as from the femur to the tibia and fibula. So these muscles are going to originate within these protuberances, within the epicondyles, within the tubercles, and they're going to attach in different areas, but a lot of them are going to attach within the surface, within the edge of each bone. That is why we have those large concentration of these protuberances towards the edges. As we will see in the tibia, sometimes those attachments are going to happen in the middle of the muscle, and you would see a line form. And once again, it's because of all of this pressure that we're putting on these. The tendons are pulling on the muscle, basically. There's as you can intuitively understand, we put a lot of pressure from the muscles to the bones whenever we perform actions, whenever we're lifting something heavy. These muscles are pulling on bones with a lot of pressure. The muscles are basically intertwined with this periosteum, this connective tissue layer that surrounds each bone. They intertwine themselves within it, and also within the ossifying areas of the bone. So that way they kind of they meld into each other, bone and tendon, bone and ligament. And that makes sense. Otherwise, whenever we do these incredible contractions, put a lot of pressure within this bone, the tendon would rip away from the bone if it wasn't intertwined completely within it. All right, lines, tubercles, epicondyles, spines. They are sharp. Their name is highly suggestive. They are sharp point projections and processes. We don't see an example of them here, but they're going to be prominent. We will look at. We will look in the cranial bone. We will look at the styloid process. Then it will help you identify. Processes are really, like spines are somewhat sharp. Processes are really sharp. You will notice a process whenever you see one. Here is these projections within the joints. So you have here the head area that helps between particular joints. You have a facet. These facets are these indentations between different bones. You have a condyle, which is basically, let's say that this facet, if these facets were in a wave-like motion, or if they have a wave-like structure, the indentation would be the facet, and the height of the wave would be the condyle. And that we see in some of your arm bones and long bones. In this particular case, we see also a ramus, which is not much of a joint, but you can kind of, in the case of the mandible, this is described as the elbow of this particular joint. Of this particular bone. It's not a joint, I might add that. Excuse me. So let's look at these different structures that allow passage of basically the piping of organisms, nerves, and cardiovascular structures. You have fissures and foramens. foramen is going to be circular, whereas a fissure it takes more of an irregular shape. It's a fissure is it's kind of intuitive. Whenever you think about a fissure of a rock, it is narrow and it's like a slit opening. Foramen is round, and a notch. A notch is an indentation at the edge of a structure. For example, here at the mandible, you have this notch present here. We also have meatus. We talk about the auditory canal. This is where those auditory nerves, the vestibulocochlear nerve, comes and goes. You also have the sinuses, these basically cavities within the bone, and we have fossas. In this view, you don't see the fossa too well. We're going to see a view of the interior of the skull that shows you this basically bowl-like shape within the skull. All right, let's look at some of the components of let's look at some of the there. I'm gonna move the camera so it looks like that we have the same field of view, that you're within my field of view. All right, let's look at the structures of bone. This is a particular long bone. This particular view allows us to see the difference between compact bone and basically this spongy bone. What do we see here? All right, let's start looking at it in more detail. It's a little busy. Let's just look at it one by one. Here you have, once again, the diaphysis and the epiphysis, proximal and distal epiphysis. So what was how do we tell a proximal from distal epiphysis? It is in relationship to the thorax, which was closer to the thorax, basically. Here we have from this proximal epiphysis, there's this epiphyseal line. We're going to look later at how bone grows, how it transitions from cartilage into calcified tissue. Here you have a view of the periosteum, this layer of connective tissue that surrounds bone. This is a highly innervated area. So whenever you break a bone, it's because of the rupture of the periosteum that you feel a lot of that sharp pain. We're going to have in the epiphysis, you're going to have compact bone for the structural stability. Here's a view of that chewy center, the medullary cavity, lined with endosteum. In lecture, we're going to focus a little more on endosteum and its role. Endosteum is going to be the layer that contains several of the main cell types that are involved in the deposition and the resorption of bone, the urosteoblast and osteoclast. It also contains stem cells that will give rise to these particular cell types. Here is the periosteum. The periosteum is going to be this layer that this is where muscles attach to, or tendons attach to, sorry. And here we have, usually each bone is going to have a main opening within the diaphysis through which the circulation comes into. And from there on, you're going to have that circulation divide or just percolate into the bone. Here you have these three structures called osteons. We're going to look at them closer in a moment. So here you have a view of compact bone more clearly. Here's the endosteum. Here you have the periosteum with these perforating fibers. These perforating fibers basically intertwine themselves completely within this growing layer of bone. The bone, as you see, is deposited in layers in different lamella. That is how the growth of these different osteums occurs. This is going to be based on this treadmill deposition and resorption of calcium based on calcium concentrations within the blood, and also based on the level, amount of pressure put on to this particular area of the bone by tendons and by ligaments or joints even. So here you have this inner cavity lined with endosteum. The nutrient foramen is going to be that original, a foramen is going to be that an oval opening within the bone. That's where this nutrient artery is going to enter the bone initially. From there on, it will travel through each bone will have, each osteon has a central canal, and that's where each one of these, the artery will branch into each central canal. So here is a cross section of bone. Here you see the arteries, veins, and nerves traveling through the central canal of each osteum. They branch off through these perforating canals. So the perforating canals do, as the role suggests, that perforate deeper into the bone itself. All right, let's look at, you're gonna hear the name lamella mentioned repeatedly, and that lamella means layers. And we're just going to describe the different types of layers that can be present within bone. So here you have the different lamella of the osteum. You would also have between each osteum, you will have interstitial lamella. You see here between each osteum, this interstitial. Between each osteum, the lamella will be present. And outside of the compact bone, outside of the osteums, of this layer of osteums, you will have the circumferential lamella that allows the bone to grow in basically thickness. Here we have a view of an osteum, and you can imagine here the interstitial lamella, and maybe this could be circumferential lamella, the ones that are on the outside, barely visible here. But here the central canal for the circulation. You see these kind of dark areas present all throughout. The bone is made out of calcium, so it's a very solid, structure that you don't think that there could be room for life, cellular life there. But each one of these darkened areas is going to be an osteocyte. The main functional cell of the bone resides within this. They reside within these openings called lacunae. So here you have a lacunae, an opening, with an osteocyte. Each one of these osteocytes communicates with other osteocytes through these canaliculi. So you see there are these radial projections visible within this tree. Those are the canaliculi that allow osteons to communicate to each other, as well as the nutrient source within the central canal. And here we have, these are the canaliculi. So these osteocytes are just basically communicating with each other, sharing nutrients and signals. Interstitial lamella, these are just lamella of the osteon, and here you have circumferential lamella on the outside of the bone. This is a view of how bone grows. This occurs at the epiphyseal plate. At some point, the epiphyseal plate will become completely compacted, and that's whenever bone stops growing, whenever we stop growing. But whenever the epiphyseal plate is still functional, this is what's happening. There is resting zone, a proliferation zone, hypertrophic, calcification, and ossification zone. The proliferation zone, cartilage, and this is what I want you to get at least at this moment, just the high level picture, is that basically we're creating cartilage, we're forming cartilage, and that cartilage is transitioning into, it's getting calcified and transitioning into bone matrix. At first, here in the proliferation zone, you see these cells dividing and migrating towards this area where they become hypertrophic. They grow in size. You can almost see the transition from cell division here to them, the cells becoming bigger and bigger. And at some point, these cells, which are still cartilage, start calcifying. Their surroundings begin calcifying. That eventually these cells die, and they will become completely calcified. This area will become completely calcified. And some cells will remain, will survive, and those cells will remain, and they will basically, their calcification will occur around them, and they will kind of create these little niches called lacunae, and those will become the osteocytes within these lacunae. All right, I've said enough. Let's take a 5-minute break. We're gonna meet at 10:15 to go over the remainder of bones. Okay, we're back. Now we're going to look at the axial skeleton. That will be your cranial bones, your vertebra, and your ribs. So let's get started. I mean, it sounds straightforward, but we're going to look at it in a lot of fine detail. So just once again, the best way to digest this information is to just constantly immerse yourself in it. All right, let's look at the axial skeleton. We're going to look at, this is giving you a view of the different cranial bones and their important markings. We mentioned the markings usually are there to reflect the function, and each one of these will give you kind of either why it's something about it or their function. Here you have the different processes, fossas, meatuses, processes, foramens, canals, foramen process. So each one of these bones will have different foramens, condyles, canals. So just try to keep that in mind. It will help you in the long run if you're able to identify the foramen, if you're able to identify a meatus, a process, a condyle. You just have to know what's the name of the specific one, and it's usually the name is attached to the function of it. All right, let's start with this particular view. Here we have the different bones. So here you have your parietal, your temporal, frontal, sphenoid, this butterfly-shaped bone, your ethmoid bone, part of your olfactory canal, olfactory cavity, respiratory cavity, lacrimal bone, nasal bone. This is your zygomatic bone, which is what we know as our cheekbones, your maxilla, your mandible. You see here different foramens already that you can imagine. It's going to be places where nutrient circulation and nerves are going to go in and out. Here I mentioned processes. Here you have the styloid process. It's going to be an open, a protuberance. Let's look at so those are the bones of the cranial cavity. I think we failed to mention the temporal and occipital lobe, but those are the bones. Be sure to know them. Now let's look at the different sutures that between these bones. So here you have the coronal suture that is kind of this is the place where the fontanelles are present. These structures that are within babies, they are still ossifying. They're still cartilage at this moment. So that the coronal suture between the parietal and frontal lobes, you have your squamous suture between the parietal and temporal bone. You have your occipitomastoid suture present between the parietal and temporal bone, specifically the mastoid process of the temporal bone. If you look, if you feel behind your ear, you will feel the mastoid process of the temporal bone. It's this very distinctive protuberance behind your ear. That protuberance is going to be sutured to the occipital bone through the occipitomastoid suture. We also have here the presence of the lambdoid suture that separates the occipital and parietal bones. So those are the sutures within your cranium that join these bones. Now, there are other structures, processes, meatus, foramens that are going to be present, or fossas. Here is the lacrimal fossa. This is going to allow the passage of the lacrimal duct to send this basically tears from the eye into the olfactory cavity. So let's look at some of these. We looked at the sutures. Let's look at some of these kind of different structures present here. You have meatus. We talk about the external auditory meatus within the temporal bone. The zygomatic process present here. As you can see, the processes are prominent. You see the condylar process present here, the zygomatic process present here. They are prominent. So I hope that you can start making that connection. Processes are fairly prominent. Here you have your condylar process, not as prominent, but prominent. Here you have your mandibular notch. We mentioned a notch is just an indentation within a bone. The mandibular ramus, or the elbow, the mandible. Here we failed to mention in the temporal bone the mastoid process present here. So basically, all of these processes allow these bones these different tendons that allow us to perform functions such as mastication, such a movement of your head. They're going to attach on these very prominent processes. This is where we have these tendons attaching to, including the condylar process. So here is the mandible. The mandible is going to have this condylar process, coronoid process, a notch, mandibular ramus, mandibular angle, and a mental foramen. The zygomatic bone, the maxilla is going to contain part of the lacrimal fossa. So that's the lacrimal bone. What else do we have here? That's pretty much it. This is a view of those markings. So I hope that you are able to, if you, for the most part, those markings, you will be able to identify them here. The sutures are prominent. The processes are prominent. The foramens are prominent. Here, the lacrimal fossa, you can see it even clearer. I guess one thing that it becomes a little blurry is the boundaries between bones. But if you look closer, you will be able to find the boundaries between the lacrimal, nasal, and ethmoid bone present here. So yeah, just go over it. Whatever we went over, just go over it here. You should be able to identify. They're fairly straightforward. All right, let's find, now this is a view from below the cranial cavity, where we can see some more of the foramen. So basically, the brain is inside this cavity. The brain communicates with a lot of structures outside of it. A lot of that communication happens through the spinal cord and spinal nerves, but a lot of it also happens to cranial nerves. Now, how do those cranial nerves get to the regions of the innervate? How do they get to the heart? In the case of the vagus nerve number 10, that is done through these different foramens. Same goes for the circulatory system. How does it get into and out of the, how does it provide the vascularization towards the brain? That is done through these different foramens present here. All right, so let's start looking at the bones first. You have the maxilla present here. that will contain a, here you have this, there's going to be, the maxilla is going to form part of the hard plate that's going to be the roof of your mouth. And this area of the maxilla present here, and also the palatine bone is going to make part of the hard palate. Here you have the zygomatic bone, the temporal bone, the vomer present here. What else do we have here? Temporal, parietal, occipital, temporal. Here is the sphenoid bone. The sphenoid bone is going to contain this greater wing present here. Those are the bones that make up the inferior view. Now let's look at some of these processes. Here we have the mandibular fossa. This is where the mandible makes a joint at this level. Here you have the styloid process. It was that really sharp process present, mastoid process, the occipital bone, and this is the basilar portion of it. From the bottom view, you observe a lot of some more complexity to the occipital bone. There is this crest that runs sagittally to the occipital bone. It ends up in this protuberance, and also it branches into these nuchal lines or neck lines present here. So there's an inferior and a superior neck lines, nuchal lines between the occipital bone, and you can imagine them being relevant for this type of movement, where you pull on the occipital bone, and then where you relax the occipital bone and pull on this area. And all of those muscles are attaching to these nuchal lines. All of those, I keep saying muscles when I'm referring to tendons, okay? Because muscles attach to bones to tendons. All right, here you have a condyle. This is a particular view of a condyle. This is here you have the different foramens. The most prominent foramens that we are going to observe are going to be the jugular foramen present here, and then this carotid canal. It looks like a foramen, but it has more of a canal, like pad, like a thicker pad, basically, to form a canal. Here's the external acoustic meatus. You have this foramen ovale, foramen spinosum, and foramen lastrum. All of these serve for the cranial nerves to move in and out of the brain. We do have a fair amount of innervation that happens in the surface of your in your face. So those cranial nerves live through these foramens and innervate your face, your tongue, and so forth. Here you have this infraorbital foramen. Here, what I want you to appreciate is the zygomatic process, how it is shaped. It leaves this opening here. All right, we're going to look from the interior of the skull. This is where I really can appreciate the different cranial fossas present. Within this bone, you have this posterior cranial fossa, this medial cranial fossa, and this anterior cranial fossa. So these basically openings or bowl-like indentations where the bone ends. At some point, before we knew much about the brain, phrenologists used to try to correlate behavior with these different indentations in the brain. I mean, if you look at bone, the relationship of muscle and bone, yeah, you can infer that there was a lot of pressure within this bone due to this muscle. However, the same principle does not apply does not translate between brain and behavior and indentations within the cranial cavities, the cranial bones. I reckon if you have a tumor for long enough and it presses against a particular surface, then sure, it might leave as an indentation, but that's about it. From there to translate it into behavior, that was an erroneous. Hold on, give me one moment. I'm back. All right. So here are the different for here. So basically, the foramen that we observe from the inferior view, now we're observing them from the interior of the skull. So here you see the auditory meatus. We're observing the external portion of it. Now we're observing the internal portion of it. Sorry, here, sorry, the acoustic meatus. Here you have the jugular foramen. Then we're going to see these different foramens here for cranial nerves, the rotundum, ovale, lacerosum, and espinosum. What else can we see here? Here we can see the cribriform plate, and all of these kind of these different foramina here, these different openings, that is for the passage of the olfactory nerves into the olfactory cavity. So if you kind of can get your bearings here, this is the interior of the skull, the brain would go here, and the cranial nerve would leave the cranial cavity through these different foramina into the olfactory epithelium. This is a clearer view of the different fossa, anterior, middle, and posterior. Anterior, middle, posterior. This is a cribriform plate. This is where the olfactory epithelium lives. Here you have the ethmoid bone. Forming the cribriform plate as well as the crista galli. The crista galli, it literally translates to the crest of the rooster. It looks like a rooster crest. Here you have the sphenoid bone. We observe here you see the greater wing and the lesser wing. So there's like this wing-like area, and here is that's smaller than the greater wing. And the greater wing, you kind of see it from the exterior, whereas the lesser wing, you do not get visibility of it. Here you have the hypophyseal fossa, and within it we will have the sella turcica. The sella turcica is basically this indentation within the within the sphenoid bone, where the pituitary lies. So the pituitary is kind of held in a even more protective. The cranial bones are there to protect the skull. The sella turcica, which is basically a little kind of a small fossa, if you will, where the pituitary lies. That kind of hints how important the pituitary is, that it has its own a small safety hole or opening for it to lie. All right, so here you have the same view. Basically, we're looking at from the top portion. This is a view from the posterior portion. From the top portion, once again, you see here the lesser wing and the greater wings. You see here the sella turcica that will serve as the resting place for the anterior pituitary. Here's the body of the sphenoid, different foramens, and this is the superior orbital fissure. Is going to be present here, so you can infer that the sphenoid bone alongside other bones are going to create that fissure. Here you have from the posterior view, so you're looking at, it's as if I'm staring at the back of this model's head. We're staring at the back of the head of this model. Here you can see the body of the sphenoid, you will see the greater wing, the lesser wing, and you will see these processes, this pterygoid process. Pterygoid. This is once again the superior view. And then posterior view. Let's look at this irregular bone, the ethmoid bone. Here you have the ethmoid bone present here. This is the crista galli, and you will see here the cribiform plate with all of its different foramina for the olfactory epithelium to make it through, the olfactory nerve to pass through this into the olfactory cavity that you see here. If there is a perpendicular plate, there are different conchas and meatuses, which basically are these bony wave-like protuberance and indentations that allow air to circulate. And then you have here these left lateral masses with which form part of the orbital plate. Here's a posterior view of the skull. I think for the most part we cover all of these, the nuchal lines, the occipital crest, mastoid processes, and so forth. In this particular view, we're going to focus on some of the markings within the anterior view of the bone. Let's start. So here, let's go over the bones: mandible, maxima, lacrimal here in green, ethmoid bone, zygomatic bone. Here you can have a view of the greater wing of the sphenoid bone, nasal bone, frontal bone. Basically, this glabella is going to be this particular portion where we frown. So wherever you feel that frown, that's going to be the glabella of your frontal bone. Here we observe within the sphenoid bone, the optic canal. This is where optic nerve number 2 comes in and out. Basically what sends signals from your visual system going in and out through here. Here is the ethmoid bone with the different concha. This is the ethmoid bone, this is the perpendicular plate, and these are the concha here. Here's the vomer. This is the orbital fissure that whenever we look at the sphenoid bone, is basically that's what I was trying to depict, the fissure present here. These are different, for the most part, we went over these processes, notches of the mandible, and where they hinge in the temporal bone. So here you see this hinge present here. All of these bones that are near the orbital area will have an orbital surface. Here you have the orbital surface of the maxilla. You also have an orbital surface in the ethmoid bone, in the sphenoid bone, and so forth. Here are the bones that form the orbit. So all of these bones will have the frontal bone, zygomatic bone, maxillary bone, lacrimal bone, and the ethmoid bone, and the sphenoid bone. All of them are going to form this opening. This is a view of the sinuses: frontal, maxillary, spinal, and ethmoid sinuses. So basically cavities within these specific bones: the frontal, ethmoid, sphenoid, and maxillary bones. Here's the hyoid bone, which contains a greater and lesser body. This bone doesn't articulate with any other bone. It's just there on its own. Here's the vertebral column. You have 7 cervical vertebrae, 12 thoracic, 5 lumbar, 5 fused sacral, and 4 fused coxal vertebrae. And so let's look at these are different maladies of it. So any deviation from the central axis will lead to scoliosis. Any deviation in the anterior through the anterior portion is kyphosis, and lordosis is a deviation towards the anterior area. This is a view of a typical vertebra. Here you have a process, spinous process, the transverse process. Here you have a lamina, the body of the vertebra, the foramen through which the spinal cord traverses through. You have a pedicle present here. and you have these facets that allow the spine to interact with the other vertebrae. So here we have a view of the cervical vertebra. One thing that you will notice is that this spinous process is not prominent. This is the spinous process, and you don't pretty much see it. And you don't see the body of, you don't see much body of the vertebra in the anterior portion either. Here, you don't see this body and you don't see this spinous process. No, so that will tell you, if you don't see a spinous process and you don't see a body, that's going to be most likely the cervical vertebra. And the less of a body and the less of a process, the closer it is to the skull, basically. This is C1. And here we have a view of C2. C2 starts having spinous process and kind of a hint of a body. C1, C2. All right, this is a view contrasting the cervical, thoracic, and lumbar spines. One thing that you will notice here is the difference between the transverse processes between the three. The transverse processes get more perpendicular and more pronounced as you go lower down the skull, down the vertebra. One thing that you will notice is in some of the cervical vertebra, the presence of this transverse foramen, and this is for the passage of these large blood vessels into the brain. The spinous process as well, you see some difference. There is not much spinous process. In the cervical, it grows more prominent in the thoracic, and the lumbar, it grows more irregular in a way. The body shape is also different. This more triangular towards the middle, the thoracic, and more round towards the. So you can see somewhat rectangular in the cervical, triangular in the thoracic, and somewhat elliptical or roundish within the lumbar areas. All right, this is the sacrum. Here you see a view of the different transfused vertebrae. These transverse ridges depict the fusion. This particular foramina allow the passage of nerves into the inferior cavity as well as the abdomen. And here we see the coccyx with these 1, 2, 3, 4 fused vertebrae. This posterior view allows us to see this crest, the medial sacral crest, present here, as well as these facets. Here, this latex, it looks like a crest. All right, let's go to the thoracic cage. Thoracic cage is going to be composed of the sternum and these ribs that are joined to this costal cartilage. Let's look at the sternum. So these are going to be true ribs, ribs 1 through 7, ribs that actually merge into the sternum, and false ribs 8 through 12 that don't merge into the sternum. Ribs 11 and 12 are going to be floating ribs. They basically do not, the false ribs merge into the sternum through this costal cartilage. They merge into the costal cartilage, and then this costal cartilage merges into the sternum. Floating ribs do not merge into costal cartilage at all. All right. Let's focus on the sternum here. Here you have a jugular and a clavicular notch. For the jugular vein, clavicular notch for the clavicle. This upper portion is called the manubrium, joined to the body through the sternal angle. And here the sternal angle is joined to the xiphoid process through the xiphi-sternal joint present here. The sternum is going to join the cavities, it's going to join the ribs through the costal cartilage. The true is going to run the ribs through the costal cartilage. Here we have a cross section of the sternum showing you the sternal angle. This is the manubrium. This is the body. This is the xiphoid process with the xipho-sternal joint. All right, let's look at how each one of these ribs joins into the vertebra. So here you have this particular rib. It has the head of a rib joining into the vertebra, which leads into the neck of the rib, which leads into this angle of the rib, which eventually leads to the shaft of the rib that joins through costal cartilage to the sternum or more costal cartilage. This is a superior view. Let's look at it closer. Okay. Head of the rib joining this vertebra. This is the neck of the rib. Here you have the angle of the rib, the shaft, joining to this costal cartilage. It's going to join the vertebra into these costal facets. So each rib will contain a superior and an inferior costal facet. And you see the ribs joining into the superior costal facet of one rib and the inferior costal facet of the rib above. Sorry, of the vertebra above. These facets are going to, here you have a superior view of this joint. The transfer process of the vertebra will join the costal tubercle of the rib through these ligaments. So here you have this ligament joining this. The spinous process has a transverse costal facet, and that costal facet interacts with the tubercle of the rib present here. So you have the transfer process has a facet, the body has a superior and inferior facet, and together those facets of the vertebra will interact with the head of the rib and the tubercle of the rib. Here we have the head with facets for articulation, and here is the tubercle that interacts with the facet of the transfer process. Shaft, angle, tubercle, neck, head, and facet for articulation. The skull of a newborn. This is the different fontanelles are just areas of chondrocyte that haven't been ossified completely. Here are the fontanelles. That's what I want you to pay attention to. And here are the different ossification centers. So this is where ossification originally arises and it spreads throughout. You can almost see the ripple of ossification that occurs. Oh my goodness. All right, that was 2 hours of bones. Go over this as many times as needed. I recommend immersing in this material in as many ways as you possibly can. As you can tell, there's a lot of different names. I hope you try to understand them in terms of their role, because every single foramen, every single meatus, every single process is there for a reason. It's there to let something through. It's there because it serves as a joint. It's there because there is a tendon attached to it that's putting pressure and making it grow. The more pressure it places upon it. Keep all of that in mind as you go through this. Do you have any questions? I'll take that silence as good. I'll take that silence as a no. Well, thank you for your time, and I hope you enjoy the rest of your weekend. And don't forget, exam number 3 and lab report, lab practical number 2 is due by tomorrow. So make sure you give yourself ample time for that.